For decades, Alzheimer's research has been divided into two massive parallel fields of study that rarely intersect. On one side is immunology. Here, researchers focus on a molecular machine called the NLRP3 inflammasome, operating inside the brain's immune cells. They have heavily validated it as a danger sensor and a potent amplifier of the disease process. On the other side is neuroscience. This field studies a large glycoprotein called Reelin, which acts as a tonic guardian of the synapse and an architect of neural circuits. We know Reelin is highly protective because of rare human genetic cases. Two individuals with mutations guaranteeing early onset dementia resisted the disease for decades. One carried a variant in the Reelin competitor ApoE, the Christchurch mutation, and the other carried a gain-of-function variant in Reelin itself, called Reelin-Kolbos. These two powerful actors are not independent processes. When you map their physics in the tissue, they form a single continuous mechanical lesion. We have simply been reading it from two opposite ends. It is a common misconception that an active inflammasome builds the toxic tau tangle directly through generalized, localized inflammation. The immune amplifier does not build the tangle with its own hands. Instead, it drives pathology by actively dismantling the structural floor that the brain's natural guardian relies upon. The physical battleground where this occurs is the extracellular matrix, the dense mesh of proteins and sugars packed between the cells. To understand how Alzheimer's progresses, we have to structurally map how an immune machine completely disables a synaptic guardian without the two molecules ever actually touching each other. The neurobiology of the disease is an architectural problem. It is a war over the physical surface that stages the brain's defenses. Inside the resting microglion, the NLRP3 machine is governed by a strict two-signal requirement. The process begins with priming. Fibrillar amyloid beta engages receptors to drive transcription of the inactive precursor, pro-interleukin-1 beta. Next is the activation signal, a rapid trigger caused by a drop in intracellular potassium. The sensor proteins oligomerize, seeding the massive micron-scale polymerization of the ASC Speck. By condensing molecules onto one platform, it forces proximity-induced enzyme maturation, converting pro-caspase-1 into active caspase-1. That single enzyme then cleaves three distinct targets, pro-interleukin-1 beta, pro-interleukin-18, and gastromin D. The cleaved portion of gastromin D travels to the inner leaflet of the cell membrane, where it oligomerizes to form a large physical pore. While a high density of these pores will cause the cell to die by pyroptosis, the pore itself is the specific conduit required for the mature cytokines to leave the cell. The inflammasome operates as a single coupled device. The pore arm delivers the cytokine arm. Now we look across the extracellular space to the postsynaptic dendrite of a parvalbumin-positive interneuron. Here we find the architect, Reelin. It does not float freely in a liquid solution. It is secreted directly into the perineuronal net, acting as a resident tenant in the dense matrix ensheathing the neuron. As this molecular model shows, Reelin requires an intact bed of N-sulfated heparan-sulfate to function. It must physically dock into the specific sulfated sugar lattice to work. This stable docking allows Reelin to successfully bind and cluster the neuronal receptor ApoE2 on the membrane below. This clustering drives an intracellular signaling cascade, specifically phosphorylating the adapter protein disabled 1. The ultimate target of this entire cascade is the inhibition of glycogen synthase kinase 3 beta, the primary enzyme responsible for phosphorylating tau.
Reelin keeps the tau protein in its normal, healthy state, bound safely to the microtubules. The brain's most powerful defense against tau pathology relies completely on the structural integrity of the sulfated sugar floor beneath it. If we pan out, we see the microglion and the interneuron sharing the exact same extracellular space. The inflammasome's primary cytokine, interleukin-1 beta, is known across biology as the most potent inducer of matrix degradation. The microglion secretes interleukin-1 beta, which commands the release of aggreganases and matrix metalloproteinases that specifically digest the perineuronal net. As the net dissolves, three things happen on a single polymer. First, the physical armor is stripped. Second, the sulfated stage is destroyed. Third, the degraded surface remodels. The stage is literally pulled out from under Reelin, lifting the brake on tau and creating the exact configuration to admit pathological tau seeds. In Alzheimer's disease, the guardian Reelin is still present. It fails because it is gagged by the immune destruction of its stage. Looking at this dual pathway diagram centered on the tau kinase GSK3 beta, we can see exactly what happens inside the neuron. Because the extracellular stage was destroyed, Reelin's inhibitory signal is gone. The protective foot has been lifted off the brake. At that exact moment, the inflammasome's second, quieter cytokine, interleukin-18, arrives through the pore. It acts directly on human neurons to upregulate the expression of that exact same kinase. A single microglial Speck utilizes two distinct outputs to target one specific neural enzyme. The inflammasome executes a perfect pincer movement. It lifts the synaptic brake by degrading the matrix with one cytokine, while it floors the accelerator with the other. Mapping this convergent lesion provides three distinct points for clinical interruption. The first intervention targets the sensor. Using small molecule inhibitors like MCC950, we can silence the NLRP3 machine entirely. This cuts off both ends of the pincer at their common source. The second intervention protects the extracellular matrix itself, sparing the sugar lattice from enzymatic digestion. This diagram illustrates a critical therapeutic trap. Heparan-sulfate has a dual nature. Specific sulfation codes stage the protective Reelin, while other codes admit harmful tau. Blunt anti-tau drugs that mask all heparan-sulfate will accidentally demolish the brain's natural defenses. The new design rule for therapeutics is strict discrimination by sulfation code. An effective drug must specifically preserve N-sulfation to keep Reelin active, while blocking the neighboring codes that allow tau to spread. Advanced Alzheimer's therapy requires surgical precision to lock the pathological gate without destroying the physiological stage. The third and most upstream intervention involves the sole human cathelicidin, a peptide known as LL37. When held in a suppressing configuration, LL37 acts as a master regulator by interrupting both signals simultaneously, neutralizing the amyloid priming ligand and blocking the P2X7 activation trigger. However, the mechanistic warning is severe. In the wrong cellular context, this exact same peptide acts as a potent activator of the inflammasome. Neither the immune Speck nor the architectural guardian initiates Alzheimer's disease. The fire is lit by amyloid elsewhere, but their collision point is where the vast majority of the amplification damage occurs. This amplification does not happen in a vague, diffuse cloud of inflammation. It happens at a highly specific physical location that can be defended. If medicine keeps the extracellular floor intact, the synaptic architect has not left the building. It can still hold the tangle at bay.